Okay, I'd like to welcome you to our second webinar. Welcome. Just a reminder, I'm Rabadan, professor at the University of Wisconsin, and I'm also the chair of the APS COVID Research and Resources Group that organizes this webinar. Just a reminder, thanks for joining. As you know, they're open to everyone, and the main goal is to bring together physicists and other related scientists. If you want to get more involved with the APS CRG, please join at Engage. And if you have any questions, there's the email crg@aps.org. Just a reminder for those who might not have been with us last time. So we have this webinars every 2 weeks. They're between 1 and 1.5 hour presentation that we'll have now is followed by the Q&A session. Then Q&A and post-webinar discussion, we move to CRG Engage platform. And then also regarding all the old webinars are at CRG Engage and they're in the APS YouTube channel video format. Few news from last time. We'll be sending the post-webinar survey. So we'll ask questions. What do you feel? What do you like about the webinar? Duration, the time. They'll also asking you for suggestion for other future webinars. So please respond to that because it's really important for us to hear what you like and what you would like to have. Also, we're looking with APS to have an additional way of signing up for webinars. There is the APS webinar platform that will allow everyone to sign for the whole series as opposed to just now that we have to get everyone individually. So this is the next 3 webinars with the dates and the titles. So you can see extremely exciting series ahead of us. With that, I would just like to thank to the, again, to the organizing committee, Professor Jan Bonot, who helps with the webinar speakers, and a lot of APS that helps with making this live. With that, I'll turn it over to Hossein to introduce our speaker for today. Oops.
Hey, thank you. So I'm Hossein Khiabanian. I'm an associate professor at Rutgers University. I'm an astrophysicist by training, but for the past few years, I've been working on computational problems in genomics and biology. So today it's my great pleasure to introduce Dr. Raul Rabadan, who is a professor of systems biology and the director of the program for mathematical genomics at Columbia University. Raul is a theoretical physicist and a string theorist by training. As you see on the website, in the introduction to Raul, he also has been a research fellow at CERN and the Institute for Advanced Study in Princeton. But for the past dozen years or so, Raul has been leading a multidisciplinary team of physicists, mathematicians, engineers, bioinformaticians, working on various problems on the statistical analysis of genomic data with a focus on infectious diseases and also tumor genomes. And he's made numerous contributions to both fields. I personally had the opportunity and the privilege of working with Raul on multiple of these problems, especially on understanding evolution of influenza and Ebola viruses when I was training for my postdoc. Last year, Raul was planning on taking a sabbatical year, but the pandemic happened, so everything got canceled. So instead, Raul wrote a book. So there's a short book that I think he's going to talk about on coronaviruses. I think, and he's also written a textbook on topological data analysis that I think he's going to talk about the applications of these types of mathematical models, mathematical tools to understanding genomic data. And with that, I think, Raul, Zoom is yours.
Thank you very much, Hossein. Thank you for the kind introduction. So let me see if I can share my screen with you. And let me see if it's okay. Can you see it? Yes?
Yep.
Okay.
Yep.
Okay. Very good. Well, thank you very much for the invitation and the introduction. As Hossein was saying, my background is in theoretical physics, but in the last 15 years or so, when I've been here at Columbia and before I was at the Institute for Advanced Study in Princeton, I've been working on problems in biology and using genomics as a tool, kind of as a lens to read some of the data. So you are probably aware that in the last few years, there have been a big development in technology that is enable us to read many genomes, human genomes, cancer genomes, and many, many different organisms, including viruses. And this is going to be the topic today. And this is going to be a mixture of two topics, something that is very recent, and this is the SARS-CoV-2. And the second one is something that we've been thinking in the last 10 years, and is how to use some tools from algebraic topology to understand the structure of spaces of genomes. So I'm going to, my talk is going to be first about the recent events, and then some of the theoretical problems that we are finding here and how we can start looking at these things from a different perspective. There's a lot of work to be done. There's a lot of data. And I think the main purpose of my presentation is that to share with you some of these problems, and perhaps, I mean, you have some ideas for helping here. Oops. Okay, so the first thing is this, I mean, people talk about COVID-19 as a virus, and COVID-19 is not a virus. COVID-19 is a disease that is caused by a virus. And one of the things that is very clear, and I think, I mean, one of the big problems, and many people, including my own group, we are working on, is trying to understand how the same virus can cause in some person be completely asymptomatic, and in other people can cause some severe disease. So having information, if we want to understand the disease, the COVID-19 disease, we have to have information both about the virus. So this is what you get if you do a PCR test, and it can give you a binary output, 0 or 1, or sometimes you get a number to send you how much virus do you have. But also it's the human and the human component. And the human is much more complex, much more complex, because it involves the history of the person, the immune history of the person, for, I mean, different viruses that we've been exposed in the past, on different pathogens, co-infections that happened. We know, for example, for many viruses, including flu, that having bacterial co-infections can contribute to the disease in a very significant way, but also the genetics. And now there are many consortia that they are compiling information about the genome of many different patients, some of those that they get very mild disease, and other ones that they get very important severe disease, even if they are usually not in the age groups of the disease risk groups. So I think this is fascinating, and we are starting to learn some of these things. But what I'm going to be talking is only about the first part. I'm going to be talking about the virus. I'm not going to be talking about the disease and how the disease is caused, but only about the virus. And the first thing is that the question that I want to address first is, where's this virus coming from and what information do we have? So for that, we have to understand how this coronavirus evolved, and then how, using genomic information, we can reconstruct how this evolution. We have many missing gaps here, many, but we have some information that I think is relevant here. So first of all, there are two different processes, two main processes. There are other ones, but I think these are the main processes. The first ones, they are mutations. So this is a virus that is an RNA virus. So the genome is a long sentence of 30,000 bases, and of 4 bases, A's, C's, and G's, and U's. And all of those, they encode the information of several proteins. So mutations accumulate in RNA viruses in a kind of almost time-like manner. They accumulate with some time, and this makes changes. So for example, in this cartoon, is changing a U into a G. And this happened. Coronaviruses, they are RNA viruses. They evolve fast, much faster than humans. I'm going to show you something in this direction. But less than other RNA viruses, they have a mechanism for proving if there's some mutations that happened in the wrong place. So here is a cartoon, or where in the space of organisms, where the genome of this virus is. On the x-axis, you have the genome size. So how big is the size of the genome? And on the y-axis is how fast it's evolved, how many mutations in a single replication gets. So per unit base. So you can see here that most of the organisms, this is in a log scale, anyway. So they are to be kind of in a line. So organisms that they are very large, like high eukaryotes, like ourselves, or a mouse here that is represented here, the genome is typically of billions of bases. And the number of mutations per replication is small. It's usually 10 to the minus 9, 10 to the minus 10, depends on the cell and the type of the timing and all things. So bacteria, they are something in the middle. They're usually the genome size is of order of a megabase, so 1 million bases. And usually the replication accumulates a little bit faster than in eukaryotes. And then we enter in the viral world that is much smaller genomes. The longest genomes in this direction, they are usually DNA viruses. They are of order of 100,000, many of those, like herpes viruses, for example. Some of those, they are very large, of order of a megabase. So they almost intersect with the bacteria. And then the mutation rates, they are much higher. They are 10 to the minus 7 or so. And then SARS-CoV-2 is here. I don't know if you can see my pointer, but it's this kind of round circle here with this saying RNA. This is what SARS-CoV-2 is. As I was mentioning, the size of the genome is around 30,000 bases. And the number of mutations per replications, typically they're in 10 to the minus 4, 10 to the minus 5 for these ones. So that means several things. And this is why it's very useful to get genomic information for these viruses. Is that as these mutations, they accumulate, and these viruses, they replicate every few hours. Mutations, they accumulate. We can use this genomic information to track down things about the origin of these viruses. So typically, the information is organized in this type of structures. They are called phylogenetic trees, where if you have, here's an example with 3 different genomes, or 4 different if you take the root. And you see the first 2 here on the right, they are very similar to each other. They only varied in the last letter. In this one here is a G, in the other one is a C. So you can deduce that these 2 guys, they share at some point some ancestor, and this is represented by an internal node. These are called the branches here. And then this one is slightly different, that probably at some point went back and had a common ancestor with the one of the node. So in some way, this is kind of representing the history of how these viruses evolve. So you can use this information if you go to this very nice place where they have tons of thousands. I mean, now I think it's almost like 100,000 genomes of this virus, and that has been collected since beginning of January of this year till now. And then you can download the genome. So we're saying this is going to be a long sentence of 30,000 bases. You can just take genomes from different places in the world and different times. There is very little information about if the person gets very sick or not, but you can get information about the physical location or where the isolate was collected. And here is a picture. It was from the first few months. This is from beginning of March, because now the structure is much more complex. And one of the things that you can do is that you just put all this information in this phylogenetic structure, where these dots represent different dates and samples. And then if they are related by a branch here, this is a short branch, that means that they are related between themselves. They are very similar, and they are more different to something that is in different branches. You can see that they are one of the things that we rapidly observed, and this is something that you can do yourself, is that you can download the data, and then you can just look at how many mutations you find. And most of these viruses, they are very, very similar. And that's important because that suggests that it's a very recent origin. So all these viruses, they are from a very recent origin. So the other thing that you can do is we can infer something about how fast they evolve using these kind of evolutionary rates. And you can get estimates of when is the common origin, the most recent common ancestor to all these viruses that has been around here. And one of the things that we observed that this happens to be kind of in October, November last year, so 2019. So suggesting that this is kind of compatible with the story that this is a new virus arriving in the population. And the first outbreak that was identified in Wuhan is probably very close to the origin of these things. So we don't have evidence that this virus has been circulating for a long time in humans. And then at some point, we were able to detect it, or people pay attention to that. Although, there could be a low level circulation that has been missed. And then for some reason, this virus took over. So the other thing that you realize, and is something that is also very clear, is here is colored by countries. And you can see that this is time is in the y-axis here. So this is from the end of 2019 till January, and then going to February again. So here now there are many more data. So I'm just chopping all this information in the beginning of March, just to give an idea, and just saying that how the phylogenetic information replicates something that we already know. So the virus is from China, most of them from Wuhan. They are all these blue dots. They are all from China, and they're very similar. You can see they are kind of diverse, but they are all kind of collected in China from the very beginning, and they are very similar. But you start seeing things, for example, that this red cluster here, this is a cluster in the United States. This is the one that was in Washington. And you can see that all of these sequences, they are all very similar, very, very similar. They are all branched in here in this particular point. So they are related to the viruses in China, but all of those, they are together. And this is suggesting that there was a single event of a migration of an infected individual with a virus, and this spread it in the community. But we can see here that this has not been an isolated event. We have many of those. So these red dots, they are plotting everywhere. So here in the US, we have many, many introductions of the virus that happens in basically here at the end of January, February, and so on. So some of those, I mean, they were able to create this community spread. Some of those, they remain kind of isolated cases. In green, light green, and yellow, you can see here very clearly, I mean, that there are some clusters here, and they are usually related to first clusters in Italy and then the rest of Europe. And they also form kind of these clade structures that they suggest that there was local transmission with several introductions. And you can see here that many of those, they are related to cases in the US, suggesting that international travel was playing a very important role here in the spread, and multiple cases coming back and forth. So this is basically something that we know. I'm going to be talking something a little bit more about what happened before these things, before December. So for that, we need to understand the second point of how this virus evolved. And it's through this process of recombination. I mean, this is different from the human recombination. It's a process where two different viruses, in this cartoon is a red and a green virus, co-infect the same cell, this here in the center, and then they are able to generate a chimera, a virus that has part of the genome is green, part of those is red, and some part of that is green again, for example. So we find that this happened in many, many viruses, viruses like retroviruses, HIV. I'm going to show some examples, and many others. Other ones, they don't follow, and they don't have these processes. They use alternative methods. But this is a way for viruses to, instead of just having like before, small changes in the genome. So like here is kind of, a small change. So if you have the space of all possible genomes, this is kind of local movements within this one. Here you can get kind of highly non-local jumps where you can have two different genomes. They can be very, very different sometimes, and they can generate something that is in the middle. So this can do amazing things because viruses and also bacteria and all these things can acquire different capabilities. So for example, if in this red piece of the genome, there is some gene that is making this virus able to do something else, or in bacterium, for example, to become resistant to antibiotics, they can acquire the capability right away by just getting this piece of material information. So this is in a process that is usually called viral recombination. And this is kind of these mutations and recombinations, they are the two main processes that we have for understanding how this virus evolved. So where do we know about where is this virus, the SARS-CoV-2 coming from? Or how these two processes, mutations and recombinations, conspire to create this COVID coronavirus? So here's one of the first papers that appeared. It was from the Lancet, where there was some of the first genomes from Wuhan. Here is a phylogenetic tree, but what they were showing before, it was basically only a part of that, only a small branch here, where now it's just we have many more, but they are all very, very similar. So you can see here. The fact that all these ones, they are basically in a bar, these are different isolates from Wuhan. That means that they were all basically identical compared to all these things. Okay. This is, and these are called Vita coronaviruses. So looking at the genome, you can see very clear this is a coronavirus, and in particular, this subtype of viruses that they're called Vita coronaviruses. There are several of those. We know many of those, and I'm going to mention three of those. The first one is one that is called SARS-CoV, and this is the virus that was the cause of the SARS outbreak in 2002 and 2003. And that was fascinating. I mean, one of the things that writing this small book, I mean, was helping me to do is just taking the time to go through all the literature in 2003, looking at this particular virus. And it's amazing how similar it was to the thing that we are finding right now. Things about even the disease, I mean, how people were getting, older people, they are getting sicker. The case fatality rate was much higher there. But how they were getting sicker, how it was more more severe for male than female. Many of these things, they were already studied in this one. So this virus, the SARS-CoV-1, is basically the cousin of this one. They are very similar to each other. They are related to viruses that they are found in bats. So in the last few years, and from different places in the world, but it has been following the SARS scare thing in 2003, there has been a systematic sampling of viruses in different species. And one of the things that was very clear is that viruses in bats, coronaviruses in bats, they resemble very similar to this one. So when this one, the new virus appeared, it was clear that it was related to some viruses circulating in bats. And you can see here that they are kind of related. They are all in the same branch. These are called sarbecoviruses sometimes. And they are mostly bats that they are found. There's a big diversity in bats. And two of those were able to jump into humans, one in 2002 and the other one in 2019. But these are not the only ones. In humans, and they have been also, since 2012, viruses coming, is thought through camels into humans. That is, one of these is called MERS, and there have been several introductions. These are viruses, they are viruses that they are not very good for propagating in the population, but they are usually causing a very, very severe disease. So there have been many jumps of these ones coming usually from people that are in close interaction with animals. And the other thing also that we have two of those that probably you've been exposed and infected before. They are common coronaviruses that are circulating around. So you have here, they are called HKU1 and OC43. So these are viruses that they are kind of circulating around. We have been sampling viruses in the last few years here in Manhattan, people that are asymptomatic. And we find many people who are infected with these viruses. Usually they are more kids, and they are usually asymptomatic. So these two viruses that are happening in the bottom here, they are kind of distant. They are also beta coronaviruses. They are in the big class of beta coronaviruses, but they are kind of distantly related, and they have been circulating for a long time in humans. There are some speculations about for how long, but this seems to be for a very long time. And they are usually not associated with severe disease unless the person is immunocompromised. So this is what was already known in the beginning of, it was I think from January, February of this year. And this is taking the whole genome of basically some of the viruses from Wuhan and some of the cousins that we know in bats and other species. And what it was clear is that this is a very recent virus and is very related to some of the viruses in bats. The closest one, there are several of those that they are close, and we can discuss this later, but this is the closest one, is one that was isolated in 2013 in the south of China, and is going by this name, RAG13. And it was found in this bat. This is a very prevalent species of bats. It's usually found in caves in the south of China and South Asia in general. And this is a very common bat. And when you sample these bats, you find a lot of these coronaviruses. So looking in the archival data, what they found is that the new virus, this one, and the virus that was found in a bat like this one, they were very similar. So they are close to more than 90% similarity in many places of the genome. So that suggests that the most likely origin of this one is from viruses that are circulating in bats. So one of the things that what we did is taking basically all the data that is here, and then trying to see if what's happened about recombination. Here is basically assuming that there are some mutations and you can just put everything into one. But we can just try to take all these genomes and trying to see if there have been different pieces of the genome that has been exchanged between these ones, these viruses. One of the things that we can reconstruct through all this data is how many of these different events with high confidence we can identify that they are kind of jumping between different coronaviruses in the beta coronaviruses. And what we found is that this recombination is pervasive. So they have been, between all these viruses, there have been a lot of events of exchange of genomic material, some of those very large. Here is a piece of the genome. So as I was mentioning, the genome is 30,000 bases. So here is basically a one-dimensional representation of the genome from position 0 to position 30,000. And these are different blocks that happen that has been exchanged between different viruses. You can see here some of those, they are very small, and you can see a small piece that is exchanged. Some of those, they can be very large, even almost half of the genome of those. If you just look at this plot, you can realize very quickly that there are a lot of events that happen in that particular region, a lot of recombination. You can just do a small plot and just trying to see how the number of recombination in a particular region is different from the uniform distribution. And you can see that there is a big jump here. And that's quite interesting because this particular region in the genome that is this prone to recombination is coding for something that is important that I'm going to mention. So just as a cartoon summary of these things is that we are finding that bats, they are usually co-infected with different viruses. And then these coronaviruses, they can recombine and they can create these new variations that usually they are asymptomatic in bats. So now what you can do is, as I was mentioning, there's a lot of recombinations in this particular region. So you can just take this particular region and instead of just doing as these guys they were doing before, is taking the whole genome, say, well, what happened if I just take the phylogenetic history of only that region? And this is encoding a particular place that is called the RBD, the receptor binding domain. And this is very important, as I'm going to mention, because that determines the host specificity. So that's a part that is basically like a key that enables a particular virus to enter a cell. So usually viruses that are in bats, they are not able to infect humans. And viruses that they are in humans, they are not able to infect other species, but sometimes they can acquire this capability through one of these processes of recombination plus extra mutations that happen. So briefly, just recapitulating some of the finding is that when you just take the whole genome and you do the phylogenetic structure, you can see, as I was mentioning before, that SARS-CoV-2 is very similar to this bat virus. These are viruses that people have found in pangolins. And you see that they are very similar, but when you look in particular in this region that determines the host specificity, here the story is a little bit more murky due to this process of recombination. And what you find here is that the SARS-CoV-2 is kind of related to the SARS-CoV-1, to the one that was given the outbreak in 2003, and is related also to these ones and some of those ones in pangolins. Another way of representing these things is through this kind of a structure. These are called tanglegrams. And what you have is that you just take a piece of genomic information and you do a tree. You take another piece of genomic information and you do a tree. And you try to see if you can align these two structures. If you can align, then you suspect that has not been any recombination. If you cannot, that means that the history of these two pieces of DNA or RNA in this case, they are not the same. And one of the things that you quickly realize is that this particular cluster becomes very similar to the one here. And these two things become very similar. So the SARS-CoV-2 and SARS-CoV, they become kind of related in this particular part of the genome, even if the rest of the genome, they are not so related. They are related, but they are not so. So and this is a very significant part of the genome. So going back, this is the part that is involved in all these recombinations. And because it has to do with the ability of viruses to enter in a cell. So for a virus to enter in a cell, it has to happen many things, but one of the things that have to happen, it has to bind to a particular protein that is called the receptor. In this particular one is in humans, it's called ACE2. It's the same one as the SARS-CoV, the first SARS. And the virus is binding here, interacting with other proteins, and then is taken to the cell through a variety of processes. Here I'm showing one through an endosome. So it's taken, and then the material is released. But so the virus, by acquiring a different receptor binding domain here, they can bind to different receptors. The ones in humans is kind of different from the ones in pangolins and the ones in other species, in bats. So in order to enter human cell, have to take this one, have to take the key and do variations in the key to enable the virus to enter. And this is what we are finding. What we are thinking that the story is happening here is that at some point that I'm going to mention, there was an exchange of genomic information between the ancestors of some of these viruses. where the SARS-CoV-1 and SARS-CoV-2, even if they're in different branches within the SARS-CoV viruses, they can exchange some genomic information and become closer in this region of the genome that has to do with the key, with the receptor binding domain. Subsequent mutations happen in both branches, and we've been exploring these mutations that lead to adaptation into the humans. Here it has been a collaboration with Mohammed Al-Qureshi and colleagues in Harvard. They've been doing molecular simulations and trying to see how these two proteins, they interact. The other thing that we can do also, because we have the phylogenetic information, we have the SARS-CoV-1 from 2003, the SARS-CoV-2 from now, and then the different ancestors, and we can reconstruct the genomes of the potential ancestor that happens here in the receptor binding domain. So from there, we have the genome, we can reconstruct the proteins, and we can see how affine they were, these proteins, to the human receptor. And this is the binding energy. And what you see is that viruses that they are circulating in bats, that they are far away, basically bind very little or anything to the human receptor. But once you start getting into the human branch, and the binding energy starts increasing, this is negative. And then here is the first, the 3A is the adaptation in 2002 that led to the outbreak in 2002. And this is the adaptation now in 2019. And here what we see is that there is kind of an increase of binding affinity that is happening here. What is also quite interesting is that some of these ancestors, for example, these ones, they already, like this guy here, the common ancestor that happened several years ago that I'm going to mention, is already have the ability to bind very effectively to human cells, to the human receptor. And that's interesting because also we can just put times into these things, because as I was mentioning, these mutations, they accumulate, and we can do an estimate of some of these times. So this is the receptor binding domain. So here we think the SARS-CoV-2 was related to some viruses in pangolin in only this region, and that was happened some years ago. They're kind of they have a small region of similarity. With the closest bat in the whole genome is this RAG13, that probably the common ancestor happened in 2010. And these are basically the confident intervals that we have. And then if you go back with SARS-CoV-1, we think that this between 1972 and 2003, this is a big gap, but there was a common ancestor where they share and this type of thing. So we have strong indication from these simulations that many of the ancestors that happen here that we can reconstruct the genomes, they already have the capability, at least to, from the point of view of binding to the ACE2 receptor, to infect humans. Suggesting also that many of these viruses that they are circulating in bats, they already have there are many viruses that they have already the ability to infect humans. That was already kind of suspected in 2003. It was very interesting when I was just going through these old papers, 2003 papers, and people were sampling in different species. It was already noted that many of these viruses, they have the ability also to infect humans. So that they could be a big reservoir of all these viruses in the wild. And when people come in close proximity, it could be through one of these wet markets, or it can be peasants that they are working in caves where there are bats and all these different things, they get infected. And then some of those, many of those, and they are dead infection and the dead end. Other ones, they can take off, and this is what we are seeing. We've seen already twice. So the summary of this part one is that coronaviruses mostly evolved through these kind of two processes, these point mutations and recombination. And on top of that, we have selection, and that is basically taking us, saying if a mutation or a recombination is going to be important or not for a particular process. Recombinations are extremely frequent in Vita coronaviruses. They are enriched in that particular region in the receptor binding domain, this particular section that is associated with the host specificity. And what we can do also with all this information is doing reconstruction of all these ancestral states. So what happened before? I mean, we can just go back and back and back. Okay, if we go very far in the past, then the uncertainty is very high. So we cannot do a lot of archaeology, but in recent things, I think we are confident that we can just try to say something. So the recombination picks up some SARS-like receptor binding domain that enables to bind to the human ACE2 receptor. So what we are thinking is that there is a binding first, I mean, this enables, and then when there is an adaptation into humans, and that there is the mutation refined interaction between these two things. The other thing that I think is interesting is that ancestral reconstruction indicates that active circulation of potentially human infecting sarbecoviruses has been at least for 20 years. So there have been very interesting studies by many groups sampling these things, these viruses, and they already saw, for example, that some peasants, they have antibodies for some of these viruses before the SARS-CoV-2. So suggesting that there is a potential reservoir and people are being exposed, and these things could happen again in the future. So this is the first part. Usually when I'm talking to biologists, this is where I stop and I give a little bit more detail into the biology. I was requested by the organizers to give a little bit more about some of the techniques that we've been using and some of those that I think they are interesting. So I'm going to describe some of the techniques that we develop. And first I'm going to motivate it going back to these plots here that I show. So you probably, if you've been following the presentation, you realize kind of a funny thing here is that when some of us, I mean, we are just taking genomes, the first thing that we do is just a phylogenetic tree, because we think that the structure of the phylogenetic tree represents in some way the history of what happened here, or how the relationship between these different things. On the other hand, I was telling you that if you just look at the part of the genome, and you took another part of the genome, you can get different phylogenetic trees. So what is exactly this representing? So it's clearly summary representation of the relationship between the genomes. But if there have been a lot of recombination, a lot of exchange of genomic material, if you take different parts of the genome, you can get very different results, and as represented here by different tree topologies that happened. So indeed, I mean, one of the classic methods that in computational biology are used for identifying these recombinations, they are changes in tree topology. So you just take different sections of the genome, you reconstruct what are your trees in there, and you see if they have been changes in topology of trees. You do some statistics and just you trust more or less the results that you find. And these are the classical methods that people are using. So some of the standard tools that are around, they are based on tree incompatibilities. But clearly, I mean, the tree is not a good representation of what is coming along, because now you can take different subsections of those, and then you can get different information. As I was mentioning, as I was showing here in all these recombinations that we found, here, I mean, you see that they are kind of not a single region that is a consistent tree history. There is a lot of recombinations. So, so now I think kind of the theoretical question is that if a tree is not a good representation of evolutionary processes involving genetic exchange, what is it? The practical problem that beyond the philosophical issues is that given a set of genomes, as the ones that you can download from GSAID, and a way of comparing them, how much can we represent the relationship without any assumption of being a tree structure? So, and there are three different features that we want to characterize in the flow of information in the system. So now we have all these genomes, and we want to understand not only how they are related in some way, but what is the type of evolution, if they are related only through mutations, or there have been some recombination of other processes. The frequency, so how often do they exchange genetic material, and in what places in the genome? As I was mentioning, this is interesting because they can tell us something about adaptation. And also the scale of exchange. So are exchanges happening to genomes that they are very similar, or they happen between things that they are very far away, from small changes, homoplacies, to something that is very large. And the other thing that we want to do is to extract features from these spaces, okay? And to perform statistics on that. So I'm going to give you a 3 slides tour in topological data analysis. This is a set of tools probably some of you are familiar with. They were developed in the beginning of the 2000s, mostly from people in the computational applied topology and also algebraic topologies. And the idea is the following. First of all, this slide is about classical algebraic topology. And I'm going to be talking only about homology groups. And the idea here is that we have an object, let's say, for example, here, a manifold, a torus, and we want to say properties about the topology of this object. So how is under stretching and all these things, properties that they don't change, and there continues the formations of these spaces. So one of those, and this is the only one that I'm going to mention, there's homology, and it's telling us how two different objects, they are related by the boundary of a higher dimensional object. So for example, here I have this particular cycle here, I have another cycle here, and they are related because there is kind of a tube that is connecting the two. So I can classify all the potential cycles up to this type of higher boundary classification. And the number of those, or the dimension of these particular groups, you can just do some algebraic invariants. They are called Betti numbers. So the BT0 is the number of points that they can be connected by a line. And this is basically the number of independent components that you have. So you have a manifold that has two different places. So you cannot have a trajectory that is going from one to another. So they are going to be two different objects. So beta 0 is counting basically the number of different objects that you have. Beta 1 is the number of holes, so one-dimensional objects. So in this case, for example, there is in the torus, there is one here in this direction and another one here in that direction. You can do the same thing with higher dimensional objects. And for example, you can do the number of voids of cavities that you can have. And this is the Beta 2. And so on. You have an infinite number of descriptors of the space. If the space is finite dimensional, then you have only finite numbers of those. So here, a few examples. So you have a single point. Well, it's very easy, it's just one point, and then there's no cycles in any other dimension, so it's everything 0. If you have a circle, you have one-dimensional object, and it's a single component, and so on. And you can start classifying these things. Okay, this is the classical algebraic topology. So we are talking about topological spaces, continuous deformations, and we can just look at the invariant properties on there. So now, what is the topological data analysis? Okay. So when we are working in the real world, I mean, in all the, when we are working with real data, we don't have an infinite number of points like we have in one of these manifolds. And sometimes the data is coming with a significant amount of noise. So imagine that you have this object here, a circle, and you can say, well, my classical invariants, this is a single component, and there's a cycle here, so these are the two. But if I am working with real data, I forget about the circle and I am doing some sampling of those with some particular noise here. And you can see here, this is fuzzy. So you forget about these things and you have only a set of points. So now the problem is if I have a sample number of, finite sample of this number of points, how can I learn properties about the underlying topological properties of the underlying space? The same thing happens here. In this case, this is a single connected component, but there are two cycles here, two independent ones, and this one and that one. So how, from this number of points, I can learn something about underlying topology. And what this is a set of tools that I'm not going to mention here in this colloquium, but this set of tools that enable you to read some properties and telling you about how much you trust the properties that you read. So I give you just the very basic example. This is the first is called persistent homology. This is looking at homology and this number of cycles. And for example, the first thing that you can do is computing the zero-dimensional homology. So this is the number of independent components that you have. So the trick that you are going to do is just you have, this is your data, for example. Here, you construct a ball of a particular size, and the size is determined here by the scale epsilon. So you have a lot of small balls here, but they're these things. If the balls and they touch, you can make a line here, an edge, okay? So one simplex. If you have three balls that they touch in the middle, then you draw a triangle. You have four of those that they touch, you can construct a tetrahedron, and so on. So what you have is that you have, for a particular scale, you have a particular, more complicated data graph, a simplicial complex that contains points, lines, triangles, and higher dimensional objects. If you increase the scale, points that were connected, they will continue to be connected, but now there are more connections. So there will be more and more connections here. And then you start increasing the scale, there will be more connections. At some point, every point becomes connected. This is basically the scale where everything becomes connected. If you continue filling up, so one of the things that you have to realize is that this object is a subset of this one in some way, because if you can always add more things when you increase the scale, but not reduce ones, and so on. And then you have this particular object. You can see here that now there were some cycles that appear at some point, some cycles, some bigger cycles that appear, and at some point they start getting filled in with higher simplices. And then so on till everything becomes a complete connected component. So now, let's say that I want to understand what is the topology of this data that I have. Well, we can just look at different scales, and I can relate the objects that I found in this scale to the objects that I found in this scale. So, for example, let's take the simple one, the number of connected components. If I take the epsilon to be almost 0, then they are going to be independent components that they are going to be all in different places. So they are going to be a lot of points here. At some point, 2 of these points become connected. And then what you do is that what is called this class dies. So there's a birth of a class and the death of a class. When they basically, now they are not 2 independent components, but the common one disappears. As you increase the scales, the number of these guys, they start disappearing, and at some point everything becomes connected, and there is a single bar that's go to infinity because there is a single connected component. So this structure here, and that is this saying how these points they join, is basically the same thing that we do in clustering when we are doing clustering with some data. Indeed, what we are doing is just taking points that they are connected and then connected again and connected again and connected again. And from this structure, the 0-dimensional structure, we can reconstruct a tree of how these things are related. But there's a lot of more interesting information here. So instead of just looking at the 0-dimensional, this is just clustering, we can just look for higher dimensions. And for example, in this case, is the 1 dimension. So you can see here that at some point there were some classes that are born where there's a cycle that appears. Oops. And at some point it gets filled in with because you can start connecting everything inside, and then it dies. So this is called the birth of the class and the death of the class. Some of those, they have very short lives, like this one. They are born and die very quickly, but some of those, they are very long. And this is the idea of this persistent homology, is that we are looking for objects that persist at different scales. So and the same thing with higher dimensional objects. So basically what this is telling us is that if we want to look at the topology of a set of points, like this one, is trivial, but we can start constructing, and there's a metric structure in these things that is going to tell us how is the relationship between these guys. Then we can start saying something about how these points are connected in different dimensions, and depends on the scale that we are taking here. If we are very small scale or bigger scale, we can set a set of objects that we can track at different scales. So how this relates to what I was mentioning before. So now let's assume now we have, for example, this, I was mentioning close to 100,000 genomes of the SARS-CoV-2 that you can download. So now you can view this set of genomes at points in a very, very high dimensional space. And you would like to learn what is the structure of this high dimensional space. If the space is really, the structure is like a tree, if there is no recombination or no funny things going on, then you can represent everything with a tree and you will be happy about that. But there could be many more complicated objects that I'm going to mention. It happens when there is recombination. So here's the same thing. Here there are 2 examples. The first is example where we don't have recombination, and we let a population evolve, accumulating mutations, and at some point we just say, well, we are going to look at the genomes. There we have all these genomes here, and then we construct the homology invariance that happened here. What we see is that if there is no recombination, we see that there is a cluster structure. They get joined with these things in 0 dimension, but there is nothing that happened in higher dimensions. So you don't see loops in this structure if there is no recombination. So the intuition here is that when we have a process that is going through just accumulation of mutations, you are basically getting this kind of branching process, and the only topology that you get is the 0 dimension, the only interesting one. However, when you have recombination, here's a simulation where we have recombinations. What you see is that you can generate genomes that are in the middle between different points, and then you start creating structures that are like that. You can start generating loops because you have information coming from different parents. So, and here in this case, in the cartoon vision of that, is that what you have is that there's a single component and everything becomes, there's a circle here because it happens because 2 different genomes, they merge and generating something like that. So that was a paper that we wrote in, I think, 2013 with Gunnar Carlson and one of my students, Joe Chan. And when we look in detail, so the intuition, if it's the data comes from a tree, that there are no loops. So the data and the data sets cannot generate non-trivial homology. So the homology in dimensions bigger than 0 is vanished. There's no petty numbers in dimension bigger than 0. And the other thing that is quite interesting also is that there are certain properties in topological. So this is an interesting field. So if you want to learn more, I'm going to give you some reference later. and that has to do with the stability. So imagine that now you take your data and you perturb a little bit because the experimentalists, they have not been sequencing very well the genome, or there can be some back mutations or something. The results, they are not changing a lot. There are some very nice inequalities that you can prove. So, so there's a very nice kind of underlying structure in these things. So here is kind of the philosophy is that you start with, in typical persistent homology, what you're looking at data that they are points in a metric space, many points. What you construct is a set of simplicial complexes, objects that they are kind of related with, in this case that I was showing with the scale. So you increase the scale, there's a set of complexes. From there, you can just do your homology algebra and compute a set of invariants that they are summarized in these barcodes. Now you can do the same thing with genomic data. So instead of just points being from sampling a manifold or something in different things, you have a very high dimensional data, and then you try to learn something about some of the invariants there. And then these are going to be a set of complexes that they are going to be related. And then the problem here, and I think this is basically the problem, is how we relate the structure that we find in these complexes to something that is interesting from the biological point of view. I'm going to give you 3 examples of how we did that. The first is flu, and then I mentioned a paper that we did with Hossein. Flu is a very interesting virus. As you probably know, we have we have 3 or 4 different strains of flu circulating right now. Flu A, there are 3. And every 30 years or so, we have a pandemic. So we have one of these viruses that they're usually circulating in birds and other animals. They are able to acquire the capability of jumping to humans, and then it's creating these pandemics. The last one that we have, it was in 2009. So we, Hossein and some of my colleagues, we were very involved in trying to understand what was going on there. So here is the structure of the virus. This is a cartoon where you see, this is a virus that is RNA, is antisense. I'm not going to discuss that, but it's a segmented virus. So it's a virus where the information is, instead of being like in the coronavirus, that is a single sentence of 30,000 bases, this is distributed in 8 different segments or chromosomes. I mean, when people in viruses, they talk about segments, not chromosomes. Each of those is containing 1 of 2 genes, and these are the names of these ones. So now, when they are two viruses like these ones, they co-infect the same cell. They can generate new combinations where one gene is coming from one parent virus and the other one is coming from the other one, and so on. So you can have many of these combinations. So we've been in the past very much interested in trying to see if there is any pattern of how these genomes are mixed. This is how we reconstructed the history of the pandemic in 2009, where we took the different segments and we tried to trace back what was the origin of each of the different segments. In this case, it's much easier than coronaviruses, because each of those, they don't usually recombine. These viruses, they don't recombine, and each of those is a discrete unit. So it's kind of, some ways kind of quantized. So you have 8 different units. So tracking this history of each 8 different units, you can just go back and then you realize that it was a mix of 2 different viruses circulating in pigs. One, it was from the closest ancestor that we had at the time. There were viruses circulating in pigs in North America, in 6 of the segments, and the other 2 segments, and they were coming from pigs circulating in Eurasia. And before that, these viruses were circulating in North America in swine. They were a mix of viruses circulating in swine, in humans, and avian. So we think that these things happen in a pig. They were able to infect several pigs, and they mix and they produce something that was able to infect humans. So these patterns of recombination, they are very, sorry, this is not a recombination, it's called reassortment. That is the discrete version of the recombination. They happened very frequently in influenza. So what happened when you applied this persistent homology thing? Well, if you just take any of the segments, so let's say, for example, one that is called hemagglutinin, what you find is that all the homology that you see is 0-dimensional. So there is no recombination within the segment, or we don't find evidence for that. You do the same thing for other segment, you don't find that recombination. So everything is dimension 0, there is nothing in dimension 1 or higher dimension. However, when you start, if you just put together these 2 things, or the whole genome, and you just put everything on those, what you find that there is a lot of 1-dimensional structures and higher dimensions that appear also in these things. So this is kind of a discrete setting with this kind of only 8 different pieces. Each of those, they can be well represented by a tree. And the structure is going to be a set of 8 trees with different histories. But if you try to take all this data and trying to infer what is the structure of these things, you will see that there's a lot of high dimensional structures. What is interesting is not just the description of the set, but you can start explaining and trying to see how they mix. So instead of taking this segment and this segment, what happens if you just take different segments? And in that way, you can just do something here and try to see if there's segments that they like to travel together. I mean, I can tell you in the previous pandemics, in the ones that we have well-recorded data from 1957 and 1968, that these ones, I mean, they happened in particular clusters of genes that they like to travel together. So in that way, you can see what genes they like to be more together in these kind of recombinations. We think that this is more associated with selection than with particular mechanisms, but we don't have the evidence for that. One of the things, they are these 4 segments that they travel together, and they are very important because they are coding for proteins that they work together. This is the polymerase. The second example is the one that I was mentioning. Now instead of just taking the whole beta coronaviruses, we go to the sarcoviruses, and then we can take different regions of the genome and trying to compute these different topological invariants, and we look at the one-dimensional ones. So if everything is consistent with a tree, what we should find is that there is no, there is no here recombination events. And what we find that there are many, and many of those in the sarcoviruses, it was compatible with what we saw before, that was this recombination that happens in the beta coronaviruses. You can do also here and just fitting a model using these topological invariants to predict the recombination rates that happen more in this region, and that is the one that is associated with the receptor binding domain. So the final example, this is one that I like, is HIV. HIV is a retrovirus, and when you just take different samples, you realize that there's a lot of recombination. There are different pieces that happened in there. Indeed, it was when I was in Princeton a few years ago, that was looking at some data from HIV. It was prompted some of the research of trying to understand what is the structure of these spaces. So if you just take this data and then you try to represent these things, you find a lot of high dimensional data where you can have these high dimensional simplices where there are different subtypes of the viruses that they are kind of surrounding the voids that happened here. So the summary of this second part is that evolution occurs in a very high dimensional space. And when we are doing genomics, we are sampling this space. One of the problems that from kind of more abstract point of view is that trying to understand what is the structure of this space from a sample of points. And then topological data analysis provides a set of tools that can enable you to capture some of these properties. So for example, you can say what is the type of exchange of genomic material that happened, and also what is the scale. So one of the things that, and the size of the bars is very interesting, is it can give you an idea of what is the scale. And also the other thing that you can do is just because you have all these bars and how the different classes happened, you can start putting statistics on trying to see how frequent they are the recombinations and use it for doing a map between the number of recombinations or rates that happened and some biological concepts and some of the topological concepts. So this is kind of the first type of the iceberg of the map between some of the persistent homology dictionary. So if you're working in the persistent homology world, and if you're working on viral evolution and how these things. So for example, we are talking about the filtration value, the size epsilon that we were saying in the balls that happened there. And that's related to some of the genetic scale that we are interested in looking at. Zero-dimensional things, for example, the Betty numbers, so the dimension of these vector spaces, has to do with the number of clusters that you have at a particular scale. The generators of the class, so the different sequences that can be combined to generate a class, is a representative element of the cluster. The one-dimensional is tell you something about the reducible recombination of reassortment events. And looking at the generators can tell you something about what were the things that happened. This is by far not done. We don't know exactly what is exact recipe here. And this is kind of basically a very qualitative difference. And the same thing with higher dimensional homology. We don't have a very good intuition of what is the higher dimensional homology means here. Most of the research that we and some of my colleagues have been doing usually stop here in the one-dimensional because it's rich enough. And here there are many interesting things. We see many examples at the HIV that you see a lot of voids and all these things, but we don't have a clear intuition with that. So I'm going to give some propaganda, and I apologize for the propaganda. This is, as Hossein was saying, when I was in the beginning of the year, that was my sabbatical. So it was a fiasco. I have all my trips, program, everything. So I stay basically not only even in my apartment, in my room. And then I was reading all the news, and because we've been working in all these vital things, so I decided just to put some of the knowledge there. It's a brief book. They have an introduction. The first and second chapter is about mostly epidemiology, giving some notions. The chapter 3 is what is a coronavirus. So all these things about SARS-CoV viruses, the common circulating coronaviruses that we have, the 4 that we have before, something about SARS and MERS and all these things. You can find it there and how it's related to many other viruses. Something that I was mentioning in the very beginning, how is the coronavirus changing? So I give a little bit more in-depth analysis of how these mutations happen, rates and things. Chapter 5 is explicitly about what do we know about the COVID-19. So chapter 6 has to do with how the outbreak of
We cannot hear you, Raul. You didn't hit the mute button by mistake, did you?
What's that?
Oh, okay. For a while we couldn't hear you, but now it's okay.
So when was this part?
Well, chapter 4 or chapter 5.
Oh, okay. So it's just local. Okay. So going back to chapter 4. Chapter 4 is how the coronavirus is changing. So things I was mentioning before about how viruses change through mutations, recombinations, and going in detail. Chapter 5 is the particular coronavirus, the outbreak that we have right now. And chapter 6 is very interesting because, as I was mentioning, I was very much fascinated by what happened in 2002, 2003 with the SARS outbreak, reading the papers at the time, and also comparing what is now with the SARS. I mean, many things, they are very similar. So these viruses, they are very similar from many points of view, but also there are some differences that are also interesting. Chapter 7 is the how it compares to flu. So there have been this metaphor of comparing these viruses to flu. And I think it's sometimes it has not very well motivated. And so I just give some introduction to flu and how these pandemics happened. And chapter 8 is basically what we know from the therapeutic options, what we learned from SARS in 2003 and what we now learn. The other one, if you are interested in topology, this is a test book. And this one, no topology. I think the idea is that everyone should be able to read it and find. And this one is a technical book. And this is basically about topological data analysis and with applications to genomics. So the first 4 chapters, they're basics on topological data analysis. The other chapters, their application. The one that I was mentioning today is chapter 5, evolution, trees and beyond. But we have many applications to cancer genomics, single cell and data, 3-dimensional structure data, and then there's chapter 9 with other applications that people have been working on. And I would like to thank many people in my lab and some people, including Hossein, that have been introducing myself, that he has been key in the studies of influenza in 2009, and some of my colleagues at the Institute for Advanced Study, University of Texas, Harvard, Stanford, Gunnar Carlson, and University of Albany, Michael Lesnick. So thank you very much.
Thank you. I guess we have to do a Zoom clap now. I don't know how that works exactly. So we got a lot of interesting questions. And so we have a few minutes. So let me get started with some of these questions then. So going back to your last slide, actually, Raul, when we're comparing phylogenetic analysis and topological data analysis, how can you find shared ancestry? Can you define shared ancestry using topological data analysis?
Yeah, that's a great question. So topology has no time direction here. I mean, so it has to be imposed on these things. When we are doing sampling, we have time because we have the isolates, what we have, for example, in viruses and all these things. So this is something that in the classical topological data analysis is oblivious because there is no directionality in these things. So we have to impose some extra conditions. So for example, if we have let's say we have a loop, and then we know that some of those, they are older ancestors and some of those, they are newer, and then we can reconstruct something there. But in general, this is true, and this is one of the things that I think is interesting. I mean, how some of the dynamic concepts can be incorporated into the language of algebraic topology.
Sorry, I was muted. Another related question is that can you somehow assess gain of function from these events? Can you understand if what the impact on the virus itself?
That's right. So what I'm describing here, so genomics is a descriptive science. I mean, what we are saying is we are looking at genomes and we are looking at the final end. So this is the product, is going to be the product of random mutations. I mean, random, that is, it can be well defined in some context. In the context of viruses, we can define very well what random means, and also selection there. So now the work of the real biology work is that when we have one of these representations, is trying to separate both. I mean, gain of functions, or looking at what is the functional consequences of a particular thing. So for example, the fact that there are a lot of recombinations in the receptor binding site. So is this due to, because there is more recombinations, is a region that is prone to recombinations, or because there's some selection for adaptation to new hosts and all these things? We don't know. So we don't know what it is. And I think disentangle the two things is an important problem. And I think basically what we have to do is just looking at all these patterns and then separating the one that is associated with selection to separate to the things that they are associated with random mutations. So the same things happen with the recombination, the reassortment, for example, that we have in flu. So we observed, and you are part of these collaborations, that some genes, they like to travel together. So is this due because there is an intrinsic mechanism of how this virus reassort, or this is due because they are having common function, and if they are separated, the virus cannot work? So we think that is the second one, but there is evidence. We don't understand very well the mechanism of how the whole virus is mixing and how it's packed. But we think that is mostly the second one. But I mean, this is the real biology where we have to do some experimental work and trying to see what happened and looking for the conditions that are doing these things.
So we have a question about infecting other people, like how virus transmits between two people. But one thing I wanted to add on top of that question, like the level of infectiousness when somebody is transmitting the virus to somebody else. But one thing I want to add on top of that, on that question, is that so these events, these recombinations, these mutations, they're happening within individuals, right? They're not happening during transition of the virus from one person to another. How much evidence do we have? Can we find exact events that these recombinations or these mutations are happening, or within an individual? Can we have you measured that, or these are speculative because we have these data from different places?
No, so I think these things have been done in vitro. So there are many papers in vitro, not with this particular virus I've seen, but in many other coronaviruses. Beta coronaviruses also, it has been done in vitro. What you can do is just co-infect a single cell with different viruses, and you can see the mixing of different things that happen.
Raul, so I have a similar question. So if you separate people by those that have very bad prognosis and those that have mild symptoms, does the virus evolve more or less in one set than the other?
Yeah, there is no evidence for any of these things. There is usually the evolution, so you can do the back of the envelope calculation. There is around, and in the book I give all these numbers, but there are around 20-30 mutations per year in this virus. So that means that it's one every 2 weeks, roughly. This is the time of infection. So it's going to be very hard to see doing some statistics because the numbers are very, very small. So it's very unlikely that you are going to pick up one of those.
Can I ask one more? If SARS-CoV was released in the human population, would it also cause disease now, or are there vaccines there?
Well, it was. I mean, so if it's going back, it's going to be very similar to SARS-CoV-2, very, very similar in many of the aspects. So there, I mean, the propagation of the virus, it was slightly different. So there were not many people who were asymptomatic, and pre-symptomatic infections, it was rare. So it happens mostly in hospitals. So there were very nice studies. If you go back, I mean, is you can just go through some of the studies in Hong Kong, Canada, and some in the hospitals that they did. And they can track basically, and then they can do antibody analysis and trying to see who really was infected or not. There were very few asymptomatic or very, I mean, that were unrealized or pre-symptomatic. It was very rare. Usually what it happens is that only people who were already sick and in the hospital, they start doing all these things. So that was one of the reasons why it was mostly confined to the hospital environment compared to this one.
We have a couple of questions about your initial part of the talk, the evolution of full genome evolution versus evolution of just the receptor domain. And going back to what you were just saying, so the severity of the disease based on, it varies obviously, but comparing to SARS, the old virus. So when you're saying that they didn't have, there were very few people who did not have any symptoms. So can we say that any other part of the genome of that virus was different from the virus that we have, and this is attributed to that?
Yes, no, that's a very good question. There is no evidence I mean, there is no functional evidence for that. I mean, analysis of severity, they are very complex. You have to do it usually in mice and doing some animal models. And you have to say, well, if I suspect that there is this particular region, I just change it, put it, and try to see if that is contributing to that. I haven't seen any systematic study of this kind of things. There are some speculations about why and the specific mutations in the receptor binding domain, they can be associated with higher binding affinity to the virus, to the human cell. There is also some using some of the proteases, some of the cells that some of the proteins that are in the cell breaking different proteins, that they are different in the two different things. How this is related to the severity is something that you have to do mostly experimentally and usually in mouse models. So that's, I mean, it's very interesting. So if you just go back also to the papers in 2004, 2005, when people were studying these things, and many of the things, for example, there are these papers looking in mice, and then, so young mice, for example, with SARS, they were not getting very sick. As they were getting older, they are getting sicker and sicker, so reproducing basically what is happening in humans, and males much higher than the females. So they reduced estrogen levels in females, and then they saw also the severity was going up. So in these systems, they were able to understand, to replicate some of the main features that you see, I mean, epidemiologically in these ones, and also to correlate with some particular molecular mechanisms, not in the virus itself, but in the host. So, and this is why there's all this interest in estrogen regulation and all these different things that happen. So I think in the next few months and years, I mean, we will have a better understanding of this question.
So I have a question. You know, part of your talk, you talked about the noise in the data that's coming into this analysis. And I'm a little not confused, but again, I just wonder how much noise is really affecting, given that, like in this case, there is like a lot of sampling. So the average noise should average out. I think when somebody makes mistakes, like in terms of the genetic mutations, that should.
The noise in these technologies is very low at the moment, very, very low. So it's like 10 to the minus 4 or 5. I mean, Hossein is an expert on looking at mutations in cancer that is much more challenging than these ones. And there, I mean, you can just go through to very low levels, 10 to the minus 4 or something like that. So it's going to be a minor contribution. When I was referring more, it was kind of back mutations and other type of things. That is kind of more biological noise in this sense.
Yep. Okay, makes sense.
That technological noise.
So maybe one question, just kind of like, how much can you predict what's going to be evolution of the virus in the future? So, you know, from this analysis that you've seen the level of recombination or added mutations. So is there a way to extrapolate future of this? You know, are it going to get more infections or not?
Well, I mean, a future. I would see what is happening, and there are certain things that they are trivialities to say. So one of those is that as the virus evolves, it's getting more diversified. So the number of mutations will increase with time. You can just plot a line, and you can see that it's getting very clear. So there is nothing new in there. Then recombinations will happen. I mean, so they will happen from viruses, different SARS-CoV-2 viruses. They will recombine. This happened, I mean, for that, you have to have a person co-infected with 2 different viruses. That we don't know how frequent it's going to happen. I mean, so this is going to be very rare. And also if this effect is going to do something about the viruses, it's going to contribute to make it more fit for some particular function, more transmissible or something. We don't know yet, I mean, these things. But I think the number of mutations will increase with time, as expected, and the number of recombinations will start appearing. At the moment, I know certain groups, and they are theoretical physicists, by the way, working on these things, and taking all this topological approach, looking for recombination in these viruses, and there are some interesting observations. But I think it's very early to say if this is going to be a significant effect or not. We know that there are 4 coronaviruses that have been circulating for very long in humans. These viruses, they probably came from an animal source. And then they happened probably in the last, I mean, not in this century, but before that. And we don't know if some of these flu pandemics that people describe historically that happened. I mean, there is this speculation that there was one in 1889, it was called the Russian flu, that could be due to a coronavirus. It was pretty bad in the whole world and all these things. So you can just go to the historical accounts. At the time, there is no isolate. We don't know exactly what caused that, but I mean, there is there's speculation that if you just take all the samples back, you can see that this kind of in the 1800s when the whole thing happened. So it could be that one of the pandemics that we thought in the 1800s or 1700s was associated with flu, it was one of these coronaviruses that came and started propagating. So, I mean, we don't know that flu was a virus till the 1930s, beginning of the 1930s.
Okay.
Jan, do you have any questions?
Oh, no, I think anyway, we are over time, so we should.
Yeah.
I think we should wrap up. I would like to thank you very much again for really interesting. I think everyone learned a lot from origins to I think how topology can help. And I think with that, I would just like to again thank you, thank Hossein for moderating, Jan for helping. At the same time, I would like to invite everyone to our next webinar, which will be given in 2 weeks by Ed Bax. And this time we'll be talking about what we know and what we don't know about the role of droplets and aerosol in transmission of SARS-CoV-2. So with that, I would like to thanks everyone for attendance. If you have more questions, let us know. We'll be moving some of the discussion also to the CRRG group. So thanks, Rabadan, and see you in 2 weeks.
Okay, bye.
Thank you.
Thank you
Bye-bye.
for the invitation.
Bye, Raul.
Thank you. Bye-bye.
Thank you.